second segment today. Back to you.
Back to me. All right. So moving on with our next speaker is going to be Stephen Hiho, who's also a PhD candidate. Stephen is completed a Bachelor of Science at La Trobe University in genetics, so he's a genetics geek, as we say. It's at the Department of Victorian Transplant and Immunogenetics Service, or VTIS as we know. It's been there for 10 years and is a very valued member of their molecular genetics team. He commenced his part-time PhD at Monash University in 2019, working directly with our team, looking at new tools for improving donor and recipient matching, and also continues his role working at VTIS as part of the on-call team, looking at they process the blood and serum from potential deceased donors and seeing if they're compatible for potential recipients. So with no further ado, I'm going to hand over to Steve. Oh, sorry, actually, I better introduce the next two speakers too. Following on from Steve will be Professor Greg Snell, who's the medical head of our lung transplant service and a professor at Monash University. He's been the head of our program for around 30 years. He's a world leader in many aspects of clinical lung transplantation and clinical research, and has championed the use of extended donor lungs to increase our lung donor pool. And he's a very highly valued member of many peak national and international donation and transplant organisations. And last but not least is Professor Glenn Wessel. Glenn Wessel is also a highly regarded specialist in lung transplantation, and also has a special interest in lung fibrosis, and is head of our pediatric lung transplant program at the Alfred as well. He has a very active interest in research and oversees our research program. He's received many national and international research grants, and he sits also on many local and national, international academic committees with regards to transplantation, and is a foundation member of the Interstitial Lung Disease Collaborative Research Consortium and a chief investigator for the NHMRC's Centre of Excellence in Lung Fibrosis. So I will now hand over to Steve to give his presentation. Thanks, Steve.
Well, thank you once again for everyone for actually having me on to talk about my research, and thank you for being part of this. As said, I'm from VITIS, so I do for 10, probably almost 14 or 15 years now. I've been working in the lab doing the HLA matching for deceased donors, for lung transplant recipients, and for other organs as well. And took a side step to do some research into sort of matching and what sort of compatibility we can use, especially for lung transplants. So first of all, I'll just talk quickly about the structure of the HLA. We do say HLA quite a lot. Some of you guys here today might understand some of it, but just to keep it really basic, there's two classes of HLA. There's HLA class 1, which consists of 3 genes, A, B, and C, which is nicely named. And then there's class 2, which is DR and DQ and DP, which gets a little bit more complicated. HLA is a cell surface protein. And as we've said earlier today, the recipient sort of acknowledges non-self HLA and sort of elicits a response. It can either be a cellular response with a direct killing, or it can be a antibody-mediated results, or antibody immunity, where antibodies develop against the donor organ or the donor HLA on the organ, and they start to kill that and destroy the donor organ, which leading to rejection, which is one of the main things we have to avoid. So HLA in transplantation. So for decades, kidney people have been using HLA for matching. They use 2 class 1 genes, A and B, and a class 2 gene, DR. So what they've found, and what has been found, is the best match HLA do much, much better than less matching. So 6, if you can see here, 6 mm relates to 6 mismatches, where 0 mm is 0 mismatches of HLA. So 0 mm is a perfect match. And as you can see there, the higher up on this chart, the better graft survival. So as we move down the mismatches, you get less graft survival. Also noting DQ is also a very bad to mismatch, but they don't use that in any of the algorithms worldwide. So my project for the last few years has been using different types of programs and ways to define HLA compatibility. There's a very out there, and I'm not going to get into detail about what each of them do, but they use a different way of defining the compatibility between the recipient and the donor HLA. Now that can be as simple as just using the gene sequence itself, or it gets to be a bit more complicated with the way the gene actually conforms, and it can form epitopes, either for T cells over here or B cells. Now, there's a few programs out there that are doing that, and we looked at all those for compatibility. Now, what we found off the top of the bat is exactly the same as what the kidneys found. That's basically the best class 2 matching goes much better outcomes. That's straightforward. We kind of almost expected that, and that was throughout the board. So what happened with class 1 mismatching in lung transplants? As I said, kidneys use class 1A and B for their algorithms. So what does it look like in lungs? As we can see here with HLA mismatches, HLA-A mismatches in the lungs, the high mismatched group does poorly in terms of rejection. Whereas HLA-B mismatches over here, the high one does slightly better, but that's not of any significance, that difference between the two. So there's something there. There's some HLA-B mismatches does a little bit better, but that's, that could be something to do with linkage with the class 2, because all HLA is linked, class 1 and class 2. But there is the other gene, HLA-C. HLA-C is not used in any matching algorithms for any organ throughout the whole world. It's sort of the forgotten gene that everyone knows exists in the labs, but they don't use it for anything at all. Now, when we looked at our data, we found something very, very surprising, where the highest mismatch group does significantly better with protection from rejection than the lowest. And as you can see here, from the high, the moderate, and the low. So it is a stepwise down. So this is very interesting. So we're seeing that the HLA mismatching at HLA-C is actually protective. That goes against 30 or 40 years of the dogma of HLA in transplantation, and it's for this forgotten gene. So what's actually happening? Well, we sort of looked a little bit deeper into it, and we know that HLA-C are used as ligands towards NK cells. So it's basically a sort of a communicating point for the receptors on the NK cells, so they can sort of talk to the donor cells and recipient cells, depending on what. So they can go past, they can screen and say, that's us, that's not us. They can protect and they can kill. So knowing that, as HLA can act as key ligands, how can we define that? So there is two types of key ligands, the HLA-C1 type over here and the HLA-C2 type. So we can look at our HLA-C sequence that we know from the recipients, and we can class them in one of these two types depending on the polymorphic positions at just two positions in the whole gene, the HLA-C gene. So using this, we looked at, as we said, we determined the HLA either C1 or C2 type ligand in all the recipients, or a C1 and C2, because there's actually two HLA-C in each recipient. And as I said, these C types actually are responsible for that direct communication with the NK cells, and NK cells can either be protectors or killers, depending on the signals from these ligands. So what did we find? We found that patients with C2 type, which we call right here C2 type, did significantly better with protection from clad. Whereas C1 type, we'll leave it as C1, and the C1-C2 type was much worse. So straight away, we can see that the C2 type patients, or the patients that express the C2 type ligand, does much better in lung transplantation. So what do we look at if we looked at the donor C type as well? So, when we always consider compatibility, we look at the donor and recipient. So looking at the donor C type and doing the same thing with the sequence and determining if it's a C1 or C2 type, we can see the mismatched C type. So that's when a donor is a C1 for recipients that are C2 did much, much better than either of the other combinations. So in this instance, mismatching of the HLA-C in specifically HLA-C type does better in long-term survival. Now, as I said earlier, could this be done through the linkage of class 2? Because HLA genes are linked, class 1 and class 2. That means that they sort of come together, and you'll see one type always in connection with another, or one HLA in connection with another. And this is a very brief graph to show in our cohort, the actual C type had more HLA-2 mismatches than either any of the other group, which is very surprising. So the theory is they should have rejected more. In fact, they had less rejection, and those that did have rejection required a significantly higher mismatch load of class 2. So they needed their HLA class 2 mismatching to be much, much higher than for the other types. So that this appears that there is a mismatched class 2 load that even C2 types can't withstand. So why? So obviously this is very early on, and we don't know exactly the process of this. This is very novel concept to say that HLA mismatching can actually benefit you. As we saw, C2 type in recipients possibly can inhibit the NK cell activity against the donor lung, but it could also be that the C2 type recipients can better fight off viruses and other infections. And if they can do that, then the immune systems are working well and they're not on hyper alert to attack donor organs. So the future directions would be to sequence the donor and recipient KIR genes. That's the receptor that these ligands actually communicate with, and see if they actually tells us what more we can infer from this. And we can also define the relationship between these KIR genes and the C-type ligands, and see if we can see something else in the data about the KIR and the C2 type, and which combinations work best and which ones work worst. And if we can find something that doesn't work well, we can hopefully try to avoid it. We can investigate the role of NK cells and the KIR in the lung transplant, and we can potentially include HLA-C in compatibility, or at least sort of widen the scope of what we can use as compatibility. Because as I said, HLA-C is not used in any compatibility worldwide, and this shows that it at least needs a bit of investigation to see what we can do with it. So in summary, the C2 type recipients are better protected from CLAD. Now C2 are the ligands for NK cells, and this goes against what we've been talked about for 30 or 40 years in transplantation. And the interaction between C2 type and donor lung is not very well understood at the moment. So we're aiming to look into this role for the HLA-C in the lung transplants, as I said, and the NK cells. And finally, I'd like to say a big thank you to my supervisors, Lucy, I'm not sure if she's online, and Glenn Westall, what we'll hear from a bit later, as well as the crew at the Alfred and the crew at Lifeblood, and to the Lungitude Foundation for their support.
Thanks so much, Steve. Fantastic. What we might do, there's a few questions, I think, coming through, but I'm going to hand over to Greg Snell now to do his talk. And we'll have some questions at the end of this session. Over to you, Greg.
Okay, thank you. I'm essentially extending Steve's work. Steve has been working with us for a number of years, and he has really produced some really interesting findings that sort of have really been translated. And I wanted to really talk about the bigger translation of things. So, and thank you, Steve, for all your work. People have been talking about matching. Well, when we talk about matching for a lung transplant, we have to match the blood group A, B, O, or AB. We have to match the height. We have to do the sickest. And then, as people have been hinting, we try and avoid the worst mismatches, but we don't really have the luxury of matching per se. We don't get enough donors in all different shapes and sizes, as distinct from bone marrow transplant, blood transfusion, kidney or bone marrow, who get lots of donors and they attempt to match. And what we're trying to match is this, the HLA has been mentioned, the immune system. And just to mention, waiting time for lungs is not that important. It's about these other parameters. And Red Cross, through Lifeblood, through Steve's team, and that is involved in the cross matching of the blood group and the HLA. So there's different types of matching the way the term's used. And the reason lungs get so much trouble is essentially here, and this is a slide of Steve's as well, that it's all about the amount of HLA you've got. And if you look down there, the amount of HLA down the bottom of there is for class 1 on the left and class 2 on the right, is the greatest for lungs and bone marrows and kidneys. And how easy is a heart or a liver up the top up there? There's not a lot of HLA, so the immune system can ignore them a bit. So no wonder we get into trouble, and the kidneys and bone marrow are matching, and we just don't have that number of organs. So essentially, and you'll see Glenn Westall there as one of those figures. There's the physicians on call are matching or mismatching. Essentially, is a bit of a gamble. We've got to get size, blood group. We've got to do the sickest. We've got to pick and choose a little bit, and we're limited into how much we can truly find a perfect match. It is a bit of a gamble the way things are currently structured. So Steve's looked at this lung transplant rejection and looked to see that essentially, what's causing it, particularly around the antibodies, what parts of can be causing troubles, and what might we do about it. And so it's basically shown that a lot of the rejection issues we're seeing are explained by antibodies in a potential recipient's blood before the transplant that are specific to the donor they might get or certainly have got. And so you can basically show that there are some bad antibodies, these donor-specific antibodies, and if there's lots of them, that's not as good. And Steve's work is carefully looked at this for lungs. And he's also further taken things that there's a subsection of lungs that he's mentioned, the epaulets, that cause most of the problem. And you've seen his most recent graphs looking at HLA-C, but over the last few years, Steve's looked at and published on the level of DSAs. If there's a DSA under or over 5,000, it determines what your long-term outcome is. If you look at the amount of epaulet matching, there's those sort of sub-fractions of the HLA that it's important. And if you put the two together, the third graph over there, you're talking of the survival of people at 10 years effectively. And you can see that you'd like to be on that blue line for sure. And indeed, if you recall back to one of Steve's earlier slides, the kidneys aren't getting 10-year outcomes like that. So you can sort of say, well, gee, if we can shift from these mismatches to getting more matches using these different techniques, we can really make a difference to people. So, have we got a new way of doing this, or at least avoiding the mismatches? And the answer to some degree is we may be able to do something. And Steve's work has basically shown that we really have to try and avoid, where we can, the pre-transplant donor-specific antibodies of a level up to about 2,000, and particularly try and avoid above 5,000. We can use an actual cross-match of the blood to give some idea of rejection, but the challenges of sending all the blood around the country, as many people have had to do, and then directly matching it in the lab, can that now be done with a virtual cross-match? And it does appear that this can be done by sorting things out in advance and making predictions as to what would be a positive cross-match. This is faster and cheaper, and indeed is now national policy. Within a couple of months, nationally, all the lungs will be done, or the vast majority of our matching will be done with a virtual computer sort of cross-match. Steve's also shown that the matching and mismatching is superior using these sub-fractions of HLA, the epaulets, to just the HLA. He mentioned that the 4 computer systems that are available to look at this matching system actually produce sort of similar outcomes. And I think we're starting to take it that if we can see that we're getting, we can predict less matches, then we may be able to use less immunosuppression in those people. And if we get less immunosuppression, we get less infections. or if there's any cancers come along, we're less likely to see that because we're on less drugs, we're able to back off, and people who've got troubles. And I think the Holy Grail is going to be going forward, and I think Steve will hopefully continue to look into this long after his PhD's finished, to define whether there's really even sub-fractions of the sub-fractions that can really tell us, well, what are the troublemakers? So some of them don't do anything at all and they're relevant, or some of them are good, or some of them are bad. So trying to understand this thing, it's not just the principle yes, no, it becomes all the shades of grey we need to understand. The Organ and Tissue Authority, which is run through Lifeblood and the federal government's put a lot of money into new cross-match systems. And again, within 6 months, we'll be getting this epaulette matching numbers for all donors for all organs. And a lot of the work in Australia has been driven by Steve showing these type of graphs that show these outcomes. So I think it's been a huge plus to be able to convince not just our sort of local colleagues, but across the country and the funders to sort of say, oh, this is the direction we should be going. Also, as part of the work that's been done is an understanding of the passenger cells that sort of leach out of the lung at the time of transplant. And it was mentioned earlier by Sandra that we sort of get a bit of an idea that some passenger cells from the lungs coming out of the donor they tell you that they're not being recognised by the recipient, and you get this sort of tolerance and less rejection. So these projects are ongoing, and both at a local level and at a national level. And I think it's very important that people sort of recognise this has really had a major impact in all organs in Australia and produced a roadmap for everyone else. And there's been lots of presentations, abstracts, and publications, and Steve and Lucy and hopefully others are going to have major career boosts out of this work as they continue to sort of to tell the stories sort of widely of what's been done. So thank you and congratulations to everyone in the team. Steve, Glenn, Lucy, Bron, Rhonda Holsworth at the Red Cross, and the supporters of Lungitude. And as I said, the work is ongoing. So thank you, everyone.
Good afternoon, all. Hopefully you can see my slide and also hear me. Thank you to Lungitude for putting together this session, and thank you for the audience for hanging on into the second hour. So what you've heard so far is very much what we're doing presently, and what I'm going to do for the next 10 minutes or so is just give you a little peek into the future and give you a sense of where we think our research directions, or some of our research directions, will be focusing on over the next few years. And I've titled this presentation From Little Things, and I'm sure many of you in this audience will be aware of the great Paul Kelly song, From Little Things, Big Things Grow. And I think that this is a mantra that very much kind of underpins how we see research. We see it really as how we take a good idea and translate it into something big, i.e. a change in our clinical practice. And that's a pathway that normally involves a middle bit, which is the research, where we test that idea before we actually put it into routine clinical practice. And I think that this narrative of From Little Things, Big Things Grow is also one that we've used to really underpin the relationship that we've developed with Lungitude over the last 10 years or so. And what we really, when we go to Lungitude each year and we talk to them about our ideas and where we want to take our research next, we often do it with this kind of narrative that we'd like to gather funding from Lungitude that will allow us to test our ideas so as to generate some pilot data and then use that funding for leverage to get more substantial funding or support. I'm going to use the next few slides just to try and illustrate that and in a little bit more detail. So, as I said, we are forever coming up with new ideas. It's basically how we improve our clinical practice with a view to improving clinical outcomes in our patients who undergo lung transplantation. We recognise that there are major funding bodies that are all competitive, that every researcher in Australia and indeed around the world applies for to get funding, and it's a very competitive field. You can approach industry to get support for clinical trials, you can approach the Australian government for Australian support, and indeed there are also international funding bodies as well. The problem is that none of them will grant funding purely on the basis of an idea. to be successful in any one of these grant submissions, you need to provide some pilot data and essentially demonstrate some proof of the idea, some proof of the concept that you're submitting for funding. And that's where Lungitude or our relationship with Lungitude has been built. We sat down with Lungitude many, many years ago and said, you know, we don't really want Lungitude to fund everything we do. What we really want to do is allow us to take our ideas along the first few steps of the research journey to create some pilot data, and then we'll use that pilot data to build major submissions to major funding bodies, and therefore leverage that initial support from Lungitude into more substantial funding thereafter. And what I'm hopefully going to show over the next few minutes is some illustrations of how that model of funding has really been very successful, and also is going to direct our research over the next few years. And this is really very much hot off the press. This is some luck we've had in this space just over the last 3 months or so. So the first area I'm going to talk about is looking at novel methods to measure immunosuppression. And this seems to be such a fundamental question, is how much immunosuppression should we give to each individual patient? We've long had this feeling that if we give, for instance, 5 milligrams of tacrolimus to 10 different lung transplant patients, the effect of that drug in suppressing their immune system may vary wildly in the actual individual. But we have no actual way of measuring that. There is the possibility that in some of our patients, 5 milligrams is not enough. And there's other patients where 5 milligrams of tacrolimus is too much. We've had a long-standing relationship with a company called QIAGEN, and they started to develop this assay called the Monitor assay. And this is an assay that allowed us to measure how effective our immunosuppression is at suppressing the immune system. So the idea we had was to approach them very early on and said, you know, could we get access to this assay and start doing a research study around whether or not it truly is able to measure how effective our medication is at doing what it's supposed to do, stopping rejection and suppressing the immune system. And we went on to do a pilot study, and in recent years that has been published. But what was critical about this study was that it could only be undertaken because we also had support from Lungitude to actually run that study, and that was acknowledged in the publication of the research thereafter. Now, what we've done with that pilot data, and this was led by Brad Gardner, who's one of our infectious diseases colleagues who does research with us in the lung transplant space. He was able to take that pilot data and put it into a highly competitive funding environment. And only just 2 months ago, he was successful in being awarded one of only 3 research grants that comes from the Major Transplantation Society. And that is to the tune of 78,000 Australian dollars. And I think it really is an example where we wouldn't have been able to do the pilot study without the support of Lungitude, but in developing that pilot data, we were then able to put that into a more substantial submission and get leverage and get increased funding for us to continue working in this space. So where else have we been successful? So I'm going to talk a little bit about tacrolimus. I've just mentioned it a moment ago. So many of our transplant patients will know that this really is the cornerstone of the anti-rejection medications that our transplant patients are given. Many of our patients will be taking tacrolimus, but it's generally taken as a tablet. We've had a long-standing interest in whether or not tacrolimus could be given in different ways other than merely a tablet. We've looked at using it just underneath the tongue, what's called sublingual. And we also described our early experience of using it intravenously. And you know, this has been a long-standing interest of the outfit of working out if there are more effective ways of delivering tacrolimus. And some of these early studies were again supported by funding bodies, and in this instance by the Margaret Pratt Foundation, which in many ways was the precursor of Lungitude. Now, many of you will be aware that many lung diseases are actually treated predominantly with inhalers. Asthma is the prime example. Asthma patients tend not to take tablets or intravenous medications, they take inhalers. So we've had an interest in whether or not tacrolimus could be used or administered via an inhaler, be more effective in the place where we want it to be effective, namely the lung, and not be associated with side effects because it's not going through the rest of the body, it's only being given into the lung. And this has been an idea that has been taken up by a major pharmacological company in the US called TFFF. And we have just signed off on a contract with TFF as the leading lung transplant center globally to run a trial looking at whether or not inhaled tacrolimus is going to improve outcomes following transplantation in our patients. And the support of that definitive clinical trial is of around the order of $350,000 to the Alfred. And again, why did they come to the Alfred to have the Alfred as the pivotal site for this international global study? Well, I think it's really very much based on the pilot work that we'd done earlier around different ways of administering tacrolimus to our patients. What was striking about this kind of story is that it took about 10 years from the very early research we did to actually lead to this company approaching the Alfred as being the lead site for this clinical trial. I mean, the final example I'm going to give is talking about some future work we're going to be doing around defeating what's called primary graft dysfunction and also chronic rejection or CLAD, which you've heard a lot about in this presentation. So just to give a little bit of background, we do our transplant on day one, and as many of our patients are aware, there's often a few road bumps along the way. But unfortunately, long-term outcomes from lung transplantation are nowhere near as good as we wish them to be. And if you look at our patients as a group 5 years on from their transplant, about half of our patients are traveling extremely well with no problems whatsoever. But unfortunately, there's far too high an incidence of chronic rejection or CLAD, which is seen in up to 50% of patients. So we have a major interest, and a lot of our research is around trying to avoid, not only recognize, but finding new ways of defeating chronic rejection or CLAD. And you saw that in all of our, and that's an undercurrent to all of the presentations that you've seen today. Now, I also mentioned primary graft dysfunction. What is that? Well, we also recognize that about 10% of our patients really start quite slowly early post-transplant, and it takes a little while for the newly transplanted lungs to fire up. And in this 10% of patients who experience this, we call this primary graft dysfunction. These patients spend more time in the ICU, they spend more time in hospital. And we're also aware that those patients who develop primary graft dysfunction as a group are also more likely to have long-term problems once they get out of hospital in the first instance. So if we can better manage primary graft dysfunction, then we may be able to also reduce the incidence of chronic rejection or CLAD. Now, what was very much a feature of the first two presentations you saw was our ability to do lab-based research on samples that we get from our patients. And for over 20 years or so, we've basically established and ran what we call a lung transplant biobank at the Alfred. And it really is a platform that underpins an awful lot of the research endeavors that we do, particularly those that are lab-based. And many of our patients who have had transplants in recent years will be aware that when they undergo their routine bronchoscopies, we're able to get a blood sample as well as a lung sample from them at the time of bronchoscopy. And we typically do that in the first 18 months or so when the routine bronchoscopies are done. And we've been able to establish really a great repository of stored samples that allows us to do our own research. And increasingly, this has been globally known about, and many companies now approach us for them to do research on the samples that we've collected historically. Now, our ability not only to run and establish this biobank has been underpinned by many, many years of research funding from Lungitude, not only to establish the biobank, but also, as you saw from the first couple of presentations, is also to fund specific research questions that we're able to ask around this biobank. And I'm pleased to announce that just in the last month or so, we have signed off a contract with CSL, who really approached us on the basis of the fact, firstly, that they knew that we had a biobank, and two, that they had some potential therapeutics that could address the issues not only of primary graft dysfunction, but also of chronic rejection. And they wanted to explore this more detail by establishing a new lung transplant biobank that will start next year and run for the next 5 years. And they have generously funded a 5-year project to establish a new lung transplant biobank that will allow them to do research and allow us to do research, and in particular to test these new therapies that they have in mind for both primary graft dysfunction and chronic rejection or CLAD. And that funding is to the tune of $2.3 million. So again, a good example where we wouldn't have been able to do any of this without the initial seed funding from Lungitude, but we've been able to leverage that to major substantial funding thereafter. So with that, I will pause, and I think I'll join all of the speakers in thanking not only Lungitude, but also their supporters. None of this would be possible without the support you give us. It allows us to translate our ideas into clinical practice. We've developed this model that allows us to get leverage of these generous dollars that we receive from Lungitude to have a lung transplant program that is bigger than we would ever be able to manage without this leverage. And in the end, it's all about improving clinical outcomes. And maybe on behalf of our patients, I'll also thank Lungitude because it allows us to have clinical outcomes and improve year on year. And with that, I'll pause. Thank you.
Thanks very much, Glenn, and everyone for fantastic presentations. So there's a couple of questions on the Q&A here about the inhaled tacro study. So maybe, Greg, I might throw this one over to you because you're the one. Does inhaled tacro hit the blood in the same way, or would it be more gentle on the kidneys?
Nicole, you've absolutely picked exactly the study design. What we're starting with, because it's the first done in the world, we're basically picking the people who've already got kidney troubles and using it as a way to back off on the total amount of tacro. So hopefully protecting the graft while running a little bit less in the blood. And that answers Jane's question as well, that it will be people they've got to be more than a year post-transplant who've got essentially for kidney indication is what it's going to be. And in the longer term, can it be done to as well as or instead of the ordinary tacro? So that's we've got great hopes that might be beneficial, and they can certainly, using a simple asthma style puffer, a powder puffer, not a nebulizer, you can get levels like you can, if you want to, you can get levels right up there of tacro.
Actually, I've got a question for Steve about HLAC. And I'm thinking already there's so much information you're trying to get for the clinicians on the night of transplant. And you know, we're going to be using this virtual matching to try and avoid known matches that were going to be bad. And how do you see, if HLAC is not being used currently, how is that going to fit into the algorithm? Would it be just for lungs, or do you think the other organs are going to be interested in it as well?
Well, it hasn't been reported in any other organ. So at the moment, it's all for us and for lungs that we're looking into it. And as part of the algorithm that you're talking about, well, I guess we can sort of predetermine the appropriate risk of potential donors ahead of time. So on the night, you'll get a sort of a low, moderate and high risk, and that will be based on the HLAC as well as the class 2 is my sort of understanding. It'll probably be a little bit more work in the lab, but for the clinicians, it should come out a bit more straightforward. It's just sort of determining at what level that HLAC does play an important role, and where we can make that decision.
I think the change in the way the government and the Red Cross are doing the cross matching at this exact point in history is perfect for this, because they're interested in development, they're interested in new ideas, and that's why we've got an advantage that the rest of the world, you know, can't do. They needed to have been forced into a change as the Red Cross has been done, and this can't be done in the US, and it can't be done in the UK as examples. This is something that I think I'm hoping we'll be uniquely proud of in 3-4 years' time.
So Glenn, I've got a question for you, or sort of a comment. I think what you showed with your overview slides there was pretty important about how difficult it is to do research and how long it takes and how expensive it is to achieve, and how important seeding funding is from organisations like Lungitude. I think too that the involvement of patients in this is really crucial, that we couldn't have done any of this without the ability of our patients to consent to participate in the studies and to allow our team to sample their blood and their BAL. I think it's quite unique, I think, compared to other organs. Do you think that the ability to actually sample the target organ so much more frequently than other transplants where they do biopsies of them? What do you?
Thanks, Brian. Yeah, there's a couple of points in there. I think that we're incredibly grateful for the support that Lungitude provides. Research is expensive. It's best done by dedicated people. That means salaries. The assays we do are often expensive, so you do need funds to be able to support it. But we are one of the most productive lung transplant research centres in the world, and that's done on a budget that is a fraction of what others in the US are able to achieve. So I think that we maximise our output based on what we get. And we had very early conversations with Lungitude, and they were very honest. They were saying, we don't want to just support everything. We want you to grow. We need to create a model that creates leverage and allows you to get further funding. And I think that model has been very successful. We have received federal funding, international funding, as well as relationships with pharmaceutical companies that are absolutely essential for sustainable research. And part of that model has been partnering with our patients. Animal models are fine, doing research in Petri dishes is fine, but developing this model where our patients have been incredibly generous with their time and allowing us to take samples from them, knowing that the benefits of the research may not be to them, but rather those who have transplants 5 years down, 10 years down the line, has been critical not only to the success of our program, but also for us to be able to attract funding from elsewhere. And we're incredibly grateful of that.
Great, Bron. I'm conscious that we're at the 1:30 mark and some people may need to leave us. So if it's okay, maybe we can move to closing the formal part of the session today, and then we can certainly stay on the line. All of our speakers have kindly agreed to stay on for an additional 15 minutes, so we can keep the question and answer piece going for anyone who has a question still outstanding or who wants to raise a question not already put in the Q&A function. So very quickly in closing, just a couple of things to update you on in terms of other great work that and deliverables that are coming up from Lungitude. So firstly, as a result of today, we'd really be keen to hear your feedback from the session. This session is really provided to give you updates on the tremendous work that the researchers are doing, and we want to know whether it is of value to you and how we may be able to improve that for the future. So we will be sending that out after this session and ask that you respond and complete that as quickly as possible, and certainly by the 17th of October if you can. We mentioned, I think, in the Q&A function before, the impact report is going to be distributed soon, which outlines all of the summary of the great research that's being done and the impacts that has in our transplant community as well. So that's another thing that's coming very soon. We've got a few other things still coming. The virtual challenge for this year. Many of you will have been aware of and involved in the virtual challenge that we had last year. This year, again, we're holding that on the 21st to the 23rd of October. And we invite you to choose your challenge to be a part of this great fundraising event, which was tremendously successful last year. And you can choose and do whatever you want, and just encourage you to register and to rally your supporters around you so that you can continue to support the fundraising activities of Lungitude. Finally, last couple of things. We have historically held a long lunch, which unfortunately we've had to delay, postpone, etc. for the last couple of years, but this lunch will still be going ahead a little bit later than planned, but in April 2023. So just mark that one in your calendar. This one is more just a chance for everyone to connect and have a nice lunch together more than anything else. And then finally, just really want to acknowledge the great work that the team do with their peer-to-peer support network. It's a great opportunity for those lung transplant recipients, their family carers to join and to speak together and to get support. It's a very big part of what Lungitude does as well. So with that, I know we're a couple of minutes over and I do apologise, but again, of course, I want to thank and call out our key supporters without whom this great work is not possible. As Glenn mentioned, the work that Lungitude supports is leveraged even further to enable bigger, larger scale research work, which can have tremendous impact on potential patients and recipients for the future. So a big thanks to our supporters, again, especially the Gillespies, the Giddis and Giddis families and Tony Pratt as well. A big thanks to you all for joining today, for your interest in this topic, for your great questions, really good questions today. I'd also like to thank the board and our fundraising committee, all of whom do this in a voluntary capacity on top of often very busy jobs. They're all driven by the same passion. We all want to make a difference to people who need to receive a lung transplant and their carers and families. And often have, many of us have a direct or indirect connection with lung transplant. So there's a real passion that drives that. And finally, I do want to acknowledge Wendy Jenkins on the call. She hasn't been on screen today, but she is such a key driver of all of the activities and has been the main person coordinating everything behind this session today. And just want to acknowledge that this year she was awarded with the Order of Australian Medal for the tremendous work that she does in lung transplant. So a very, very big thanks to Wendy as well. So with that, we close the formal part of today, and apologies that we've run a little bit over. As has been mentioned, the recording will be available, and I believe the link will be provided to those who have signed up for today's session. If anyone wants to reach out separately, please feel free to do so at any of the contact points on the screen at the moment. And for those who wish to, please stay on for some additional time with our tremendous speakers, again, whom I'd like to thanks for their, not only for their presentation today, but for their dedication to this really important area of research. So big thanks to all, and thanks, Bron, for the co-host extraordinaire today as well. And many thanks. We didn't share the poll outcomes today, Wendy, but I don't know if you want to quickly flick those up on the screen as we farewell people from the call today. But I'm assuming we've obviously got some people joining us from Sydney. But there we go. You can see hopefully on your screen the majority from Melbourne, but we do have some regional Victoria attendees, South Australia, New South Wales and elsewhere. So I think that's the value of the virtual connection. And most people on the call today are either directly connected as a recipient or health professionals. So many thanks for answering the poll and for joining us today. So we'll close the formal part and then just move across to any additional questions for those who wish to stay on the line. A big thanks. Thanks all.